This is the first topic of Unit 2, Prokaryotes. Unit 2 also includes eukaryotes and viruses. Warming up with prokaryotes, you might want to pause your screen and take a moment to fix this paragraph. There are several things wrong with this paragraph. And here are the answers. Lactobacillus was not written correctly, genus and species. The genus was not introduced. It was not abbreviated properly. Bacteria is plural. Bacterium is singular. And then lower in the paragraph, when it's talking about lactobacilli, that's a common name. That's not a Latin name, and it does not need to be capitalized and italicized. Now, in our last unit, we did some light overview of the scope and history of microbiology, and we talked about the differences between prokaryotes and eukaryotes, and how now there are 3 domains instead of 5 kingdoms. But we still often group bacteria and archaea into prokaryotes and all of eukaryotes together so that we can make comparisons. And of course, prokaryotes are smaller than eukaryotes, and the big one is that they don't have a true nucleus. Well, a true nucleus is a membrane-bound organelle, and there are actually lots of membrane-bound organelles that eukaryotes may have that prokaryotes do not, and that includes the nucleus, mitochondria, chloroplasts if they're photosynthetic, the Golgi, the ER, and vacuoles. Also, there's a difference in the way that their DNA is packaged. The number of chromosomes, prokaryotes only have 1 chromosome versus multiple for eukaryotes, and prokaryotes may have plasmids, which we'll talk about again later. Because there is no nuclear membrane dividing the DNA from the cytoplasm, in prokaryotes, transcription and translation are coupled. It's often called co-translation. Whereas with eukaryotes, DNA must be transcribed into messenger RNA, sent out through nuclear pores into the cytoplasm or the rough ER, and translated into protein in the rough ER. Both organism types have viruses. In fact, all organisms have viruses. There are viruses for humans, animals, plants, and even bacteria. So prokaryotes are smaller than eukaryotes. They are the smallest living cell, although viruses are even smaller, and eukaryotes are often at least 10 times bigger than prokaryotic cells. And if you remember this picture that I showed you from our very first day, starting with a flea, which we could see, and filamentous fungi, we have protozoans at the top, and then mold spores, and then we get into spirochete bacteria, bacillus bacteria, cocci, ricketsias, which are intracellular bacteria, so they're very small, and then we get into viruses, which are even smaller, and all the way down to DNA and protein. So, cell shape and arrangement. Now, there are actually lots of cell shapes, but this was the most simplistic picture that I could come up with. On the far left, the rod-shaped cell is called a bacillus, B-A-C-I-L-L-U-S. And multiple bacillus are called bacilli. The next one appears round on a 2-dimensional screen, but it's actually a sphere. Spherical bacteria are called cocci, that's plural. If you have just one, as we've pictured here, that's a coccus. And then the next 2, the curly ones, the ones that are kind of spring-shaped, are spirochetes and spirillum. Spirochetes and spirillum really only differ in terms of length and where they're found. Spirochetes are often ones that we see in medicine, whereas spirillum are aquatic and usually avirulent. The only distinction here that you need to know is that they're curly because they have periplasmic flagella. You don't need to know the difference between spirochetes and spirillum. And then lastly, the comma-shaped one, or the C-shaped bacterium, that is called a vibrio. You can have one vibrio or many vibrios. Now arrangement. Arrangement has to do with the way the cells divide and whether or not they separate from one another. Most often there is no arrangement with bacteria, but if there is a specific arrangement, that is something notable that we can actually use to help identify the organism. And so if an organism divides, it can be called a diplococcus in this case. And if the organism is only in pairs, that's diplo. Whereas if it divides into many pairs forming a string, as in over here on the far left, you would see a strepto. That's a strepto arrangement when it's a long chain. Now, because cocci are spherical and they divide by binary fission, but that plane of division can be anywhere because it's a sphere, they may not separate and they may not divide in any particular string or order, and so then they end up forming clusters. And clusters is called a staphylo arrangement, staphylo. So in this case we have a diplococcus, a streptococcus on the far left, and a staphylococcus in the center. Staphylococcus. I remember the difference between strepto and staphylo because bacteria are gossipy, and your staph bacteria clusters around the water cooler to gossip. Staph clusters around the water cooler. And if you can remember staph clusters, then you'll remember that strepto is in chains. Now you can find diplobacilli, or you can find streptobacilli, like pictured here on the far right. However, you will never find a staphylobacillus. You can never find a staphylobacillus because, as I mentioned, they divide and separate. And so while a coccus can divide on any plane because it is a sphere, bacilli will only divide on the skinny plane. They will only divide on the shortest part, never long ways like a hot dog, and certainly not at some angle. And because they can only divide along the shortest plane, that means that they can only form no arrangement, diplo, or strepto. They will never be staphylobacilli. There are other arrangements, such as tetrads and sarcina. These are often found in soil microbes, very interesting. We don't often see those with medical microbes. And truly, when we talk about cell shapes, we tend to draw them very uniformly, but microbes come in all kinds of cell shapes. Cocci are not always perfectly spherical. Bacilli are not always perfectly hot dog shaped or brick shaped. Sometimes they can have oblong ends or tapered ends. And we even have some organisms, such as Corynebacterium, that look like baseball bats, or Streptomyces is a type of bacterium that looks like a mold, but it's truly a bacterium. Molds are fungi. Fungi are eukaryotes. But these ones are very filamentous, and even on a petri dish they often look like a mold. And so even though we talk about these in very distinct groups, we often can see different shapes. So here's scanning electron microscopy of different shapes of bacteria. See if you can pick out the cocci, the bacilli, and the spirochetes. Now, can you see an arrangement? There's often no arrangement, so that's not a surprise, but there are diplococci and there are diplobacilli. Okay, test for you. Is this a coccus, a bacillus, spirillum, or spirochetes? It's a bacillus. Next one. Cocci, bacilli, spirillum, or spirochetes? These are also bacilli, but they are in a strepto arrangement, so don't be confused by the kinks between the cells where the two cell ends meet. This is not a spirochete or a spirillum, this is strepto or diplobacilli. Next, cocci, bacilli, spirillum, or spirochetes. These are cocci, these are diplococci. This is a capsule stain, and this particular organism is Streptococcus pneumoniae, and it causes pneumonia because of that capsule, and it's often found in a diplo formation, even though it is a streptococcus. This is the same, Streptococcus pneumoniae. There's another one. Now, we have to look, what are we looking at here? We're looking at human cells, neutrophils mostly, white blood cells, coming to the site of an infection. So you have to spot the bacteria. If you spot the bacteria, remember they are much smaller than eukaryotic cells. Yes, there's a couple here, and there are several over here. These are diplococci, diplococci, diplococci. And what is this arrangement? This is a strepto. These are in chains. Yes, they are in clumps. However, that is not a true staphylo arrangement. If we were to see a true staphylo arrangement, it would look as if a little kid were drawing grapes for us, a picture of grapes. It's very uniform and very geometric looking. And if you ever see long chains like this, that is definitely a strepto arrangement, and they may be just clumped or piled on top of each other. This is a bacillus. It is in a strepto arrangement. This organism is Bacillus anthracis, causes anthrax. It's often found in a strepto arrangement, and it doesn't gram-stain properly because of unique lipids, so that's why there's some variation in the color. And then lastly, this is a filamentous bacterium, one of those mold-like looking microbes that is actually a bacterium. Okay, so we talked about cell size, shape, and arrangement, and now for the next part of this topic, we're going to talk about the organization of a prokaryotic cell. And this is your roadmap. We're going to start from the outside and work our way towards the internal structures. When it comes to external structures, we're going to start with flagella. If you have many of them, it's flagella, such as bacteria. And if you have just one of them, it's flagellum, such as bacterium. Now, because there are two types of bacteria, which we'll get into, gram-positive and gram-negative, there are two types of anchors that seat the flagella in the cell envelope. But all flagella have an anchor, which is this stuff down here at the bottom. And all flagella have a curved hook and a filament, and this is the long solid protein that makes the actual flagellum. Now, when we're talking about flagella, if an organism has a flagellum, that is notable. That's something that we can use to help identify it. And if we can do a flagellar stain, and we can look at the arrangement of that flagellum, again, that is more information that we can use to identify it. So, on the top here, If I have a flagellum and it's just one flagellum, I call that monotrichous. Mono meaning one, and trichous, they're all going to have trichous on them. Trichous, monotrichous. If they are in a tuft, that's lophotrichous. Lophotrichous. If they are in tufts or singles, but they are on both polar ends of the microorganism, so this must be a bacillus, in this case, at polar ends, then that's called amphitrichous, A-M-P-H-I, trichous. Amphi, I remember this one because mono means one. Amphi, if you can use both your left hand and your right hand, you are ambidextrous. So ambi sounds like amphi. And then this last one goes all the way around the perimeter of the organism, all the way around the perimeter, or peritrichous. Peritrichous. Now remember that those flagella always have an anchor, a curved hook, and the filament protein made of flagellin. That curved hook spins, and that's how a flagellum works. It spins by rotating that flagellin protein in a corkscrew motion. So it's very much like a propeller on a boat that spins in a corkscrew fashion and pushes the organism through the aqueous material. Now that's called a run when the organism is moving, but organisms may need to change directions or pause in order to sense their surroundings. In order to do that, they reverse motion. So if they're going, in this case, counterclockwise to run, then they would reverse it to clockwise rotation. And when you reverse a propeller, it stops motion for a little bit. And all the flagella will fling out, and the organism kind of floats in the aqueous, and it may continue to work in a clockwise motion and go in the opposite direction, or it may continue in the counterclockwise motion and then again continue on its original track. This entire progress, while not perfectly linear, run and tumble together, is called chemotaxis. Chemotaxis, when we're talking about bacteria like here or any other cell, is by definition the movement in response to a chemical gradient. So this organism may be moving towards it or away from it. Altogether, that movement in response is called chemotaxis, and it's a series of runs and tumbles. Now, just for funsies, would you expect a bacterium such as E. coli to have a flagella or many flagella? Think about where E. coli lives. E. coli lives in the colon. Would you expect a colon-dwelling bacterium to have a flagellum or many flagella? Yes, it does. Absolutely needs to be able to move around inside the intestines and respond to those chemical gradients. Here is another electron microscopy image of a bacterium or an Escherichia coli. And what would you say that arrangement is? It goes all the way around the perimeter. It is peritrichous. Peritrichous. And which one was lophotrichous? Lophotrichous. B is lophotrichous, yes. Mono is 1, and amphi, as in ambidextrous, is C. Now, there are always weirdos in every case, and I mentioned to you that spirochetes and spirilla are shaped like corkscrews because they have periplasmic flagella. Periplasmic flagella means that between the cell membrane and the outer membrane, those corkscrew flagella are inserted, or that's where they reside, which means that the whole organism itself works like a propeller because those flagella slide back and forth across one another, turning the whole organism. So why are spirochetes and spirilla shaped the way they are? Because they have periplasmic flagella. Now, one more significant note: if an organism has periplasmic flagella, if it has a large enough periplasm and it has an outer membrane instead of a thick peptidoglycan layer, it must be a gram-negative organism. So all of our spirochetes and spirilla are gram-negative. So why do we care about all of this? Well, I mentioned it is important to identify organisms. It helps us get one more piece of information that would tell us about the bacterium. Those flagella also function in microbial virulence. They can work as adhesion and immune suppression. And also, they are a great target for antibiotics because we, as eukaryotes, do not have bacterial flagella. We don't have flagellar protein, flagellin. We don't have that anchor and that hook. And so if we could give a person a drug that would disrupt the assembly or production of a flagellum, it could kill the bacterium without harming the host. Next up in our external appendages are pili and fimbriae. Now when it comes to pili, that's plural, or pilus, singular, your book may discuss pilus or pili being used for attachment. There are some pili that bridge the gap between fimbriae and pili, and so there is some confusion about that, but we are going to focus on the specialized structure of a pilus used as a sex pilus. And it's an elongated, rigid tube, so it's hollow, made of protein. It's only found in gram-negative bacteria. And it's also only found in gram-negative bacteria that have a plasmid, a special extra chromosomal piece of DNA that encodes the genes for making a pilus. Now, what is a pilus for? If an organism has this, it can make this long hollow tube and project that pilus out towards other bacteria in the area, and then that plasmid can be transferred via the pilus, which is why it's called conjugation, because it is an exchange of DNA.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. A plasmid is a small piece of DNA, separate from the main chromosome, that carries genetic information for such things as antibiotic resistance. The first step in plasmid transfer is contact between the donor and the recipient. The pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when an enzyme cleaves one strand of the plasmid at a specific nucleotide sequence, called the origin of transfer. A single strand of the plasmid, beginning at the origin of transfer, enters the recipient cell. Once inside the recipient cell, a complementary strand to the single DNA strand is synthesized. When donor and recipient cells are mixed together, eventually all of the cells become donors.
So you can imagine that when a cell is completing conjugation, it must be a gram-negative cell, because if that pilus were to come out and meet a thick peptidoglycan mesh network, that hollow tube would not be able to penetrate that. Whereas if it were to meet a phospholipid bilayer of an outer membrane, that has a fluid mosaic model. It would shift, it would flex, and it would be able to accommodate that hollow yet rigid tube. So only gram-negative organisms perform conjugation. Only gram-negative organisms with an F plasmid can make a pilus to perform conjugation. Next are fimbriae. Fimbriae are short, bristle-like fibers on a cell that help with host attachment. So not these long ones, those would be more like Peritrichus flagella, but rather the short, hairy-looking ones. And they help with attachment to hosts and each other, such as pictured here, as well as other surfaces. So fimbriae are part of attachment. So that was the appendages. Next up is the glycocalyx. And when we talk about the glycocalyx, there are two types. There is a slime layer and the capsule. They are all made of sugars, long-chain sugars, polysaccharides, but the difference between a slime layer and a capsule is that one is more loose, so it's easier to remove, and one is much thicker and more tightly bound. They both protect an organism from dehydration, from drying out, and because they are long-chain sugars, they can be reeled back in by the organism and used for food, so they can prevent loss of nutrients. And they are used for adhesion as well as protection. Now a capsule I mentioned when I talked about Streptococcus pneumoniae and that capsule stain. A capsule is often easy to spot on a petri dish because it protects the organism from dehydration. And so we could look at just the colony morphology of the organism and it would appear mucoid, wet, runny, drippy, snotty, because that long-chain sugar capsule is holding moisture in around the organism, and you can actually see that moisture collected around the colonies on a petri dish. So these are capsule stains, and you can see especially over here in the middle left that the capsule, the sugars themselves do not stain, so we have the background stained and the bacterium stained within, and that is a way we can look microscopically and tell if an organism produces a capsule. Now, fimbriae and capsules or slime layer are important appendages or important external structures used for the formation of biofilms. Biofilms are groups of organisms, so many types of organisms. It does not have to be only bacteria, and it does not have to be one type of bacteria. It can be bacteria and eukaryotes of several types, forming a slimy layer in an aquatic system. So what they do is they use fimbriae to attach to one another and to surfaces, and then they build up a thick layer of glycocalyx, slime layer, and capsule in order to bind everyone together and to that surface. This is almost always aquatic, but it could be in nature, like rocks and streams. It could also be in humans, like your intestines have a thick biofilm layer. It could also be in things such as catheters, which is why we have switched now to disposable catheters, because that thick layer of glycocalyx protects the organisms from drying out. It protects them from applications of disinfectants and antibiotics. It protects them from heat and starvation. And in addition to all of that, what are they doing in there? they are benefiting from one another's enzymes. Perhaps you can break down sugars that I can't, and when you break them down, I can eat the crumbs. Perhaps they are performing conjugation and swapping genes and gaining new things such as antibiotic resistance within those biofilms. These biofilms can also form in transplants and joints, but you might be closer to a biofilm than you think. I told you your intestines, but every night when we brush our teeth, we scrape off our biofilm, and when we clean the toilet, we are cleaning off a biofilm. And if you have a dog's water dish and you've ever tried to clean that and it's just slippery beyond slippery and it never seems to come clean, that's because there's a biofilm formed in there. So biofilms are found just about everywhere, and they have lots of benefits and some things that are not so good. Next up, we'll talk about the cell envelope. I'm going to put a pin in this video for now and pick it back up again there.